Live now. Okay, well, good afternoon, everybody. Good afternoon here in Oxford. Good morning to those of you in North America. My name is Rene Bobe, and welcome to Primate Conversations. It's a real pleasure today to have Bonnie Jacobs joining us from Dallas, I presume, Dallas, Texas. Is that correct?
Yes.
And sorry, I get some feedback there. So back to our presentation. It's a real pleasure to have Bonnie Jacobs with us. Bonnie, I think, is probably the foremost paleobotanist working in Africa today and for the past several years. I know that she and I have met in Kenya at certain points and Ethiopia, but Bonnie has a very long history of research in the evolutionary history of African ecosystems and vegetation, mostly through studying paleobotanical remains. She's an emeritus professor now at SMU, Southern Methodist University in Dallas, and has a BA from SUNY Buffalo, which is a place where I spent a couple of years as a assistant professor, and a master's and PhD in geosciences from the University of Arizona. And Bonnie has a very long list of very important publications. And for those of us interested in the evolution of African ecosystems and how faunas and mammals and various types of organisms have evolved over time in Africa, her work has been very, very important, both in describing specific sites as well as providing the big picture, which has got some very important publications sort of about the origin of grasses and the vegetation history of Africa. And again, her work has been critical for those of us interested in the evolutionary history of Africa and its fauna and ecosystems. So Bonnie, thank you so much for joining us, and I leave you to it. And for those of you who may have questions for Bonnie, please be sure to write them in the chat function in YouTube, and then we can provide those at the end of the talk. So thank you for being with us.
Renee, thank you so much. Thanks for this invitation. It's really very special to me, and I want to also thank you for that lovely introduction. It's really nice. Okay, so let's talk about, I thought it would be best actually to look at some things in detail rather than to do a big overview. So my screen is not, there we go. Rather than to do a very broad brush overview of things, because that can get us to more specific examples of paleoecological approaches. And I thought that maybe that would be best for this talk. I hope so. So the goals here today are to convey African vegetation categories. First of all, what is it that we're trying to reconstruct? And of course, vegetation in Africa, it can be classified at multiple scales, just like every place. And then we have to keep in mind, once we understand a little bit about that, how is it possible and how do we go about getting to that kind of a level of reconstruction? What can we actually do with not just plant fossils, but plant fossils plus all the other proxies that we might want to use? So then I'll briefly provide examples of microfossil and macrofossil source area and taphonomy, mainly in lacustrine settings, because the example that I'm going to talk about from the early Miocene is a lacustrine setting. So at least we can narrow taphonomy down to that for this case. And then I'll report on our proxies of paleoecology at this early Miocene site called the Mush Valley, which is northeast of Addis Ababa on the road to Dessie, a beautiful place on the edge of in the uplands, but on the edge of the rift. So first, let's do some defining of ecological terms for vegetation units, from largest to smallest, and I'm not going to cover every single one. That's a whole course in itself, but to get an idea of the complexity out there and actually the challenges that ecologists have in classifying vegetation units. There's a new, well, relatively new resource for people, and it has to do with ecoregions, which is probably not a term we're used to using, but they seem to be very handy because they include both plants and animals together, and they're at a scale that might be useful also for people doing paleoecology. So I'm going to interject that in here. But first, some terms. So a biome is a very large scale kind of classification. It's groups of plants and animals occurring in particular climatic and geographic areas. So climate is the main underlying thing that unites biomes. And so you can see right here in this map, the colors represent different biomes. And say the green here, for example, is showing the tropical forest biome across Africa. And you can see that the green picks up again on the east coast of Africa. And that's because in the middle there, that orange color represents a different biome because the climate is different there. It's a drier climate. Why? Because of course, that's where the rift goes through. So that's what a biome is. It's pretty broad. An example is the tropical and subtropical moist broadleaf forest. Okay, that's the green. And then ecoregions are relatively large areas containing distinct assemblages of plants and animals, smaller than a biome, but usually bigger than a single community. And these are roughly equivalent often to Frank White's classic publication, The Vegetation of Africa, which is still absolutely useful and important. And so he has things called floristic regions of Africa. So that's sort of roughly equivalent to these ecoregions. And another example would be the central Congolian lowland forest. So that's a part of this big green forest biome. And then communities are just associations of interacting populations or species, usually defined by how they're interacting, but often they're named by the key compositional elements. So for example, here in Texas, we might have the oak hickory forest. So when you're in an oak hickory forest, you're in that kind of a community. And communities are important for paleoecology because oftentimes what you're sampling is from the local community. Of course, that fits into some ecoregion and some biome, but what you're really seeing with your fossils is the local community. So an example of that would be mixed, moist, semi-evergreen rainforest, which is another subset from White. And I wanted to show you this interactive map very quickly. So I've touched on this. I need to share my screen again, a new share. Let's see if I can do it. Okay, yes. Okay, so Megan, you tell me if I'm not sharing the screen. Okay, so here are these ecoregions, and this resource is really nice because this overlays the vegetation units onto the Google Maps satellite imagery. So basically you see a more direct reflection of reality from a satellite, right? And then you can overlay on top of this the biomes, which is what we looked at before. And now here are the ecoregions. And you can then get some more information about each ecoregion. So for example, here, that central Congolian lowland forest, which is part of the tropical, subtropical, moist, you're never going to remember all these things, but, basically as a part of the big tropical forest that goes across the wettest, warmest parts of Africa. And so here's a lovely picture of it. So it includes animals and plants. And you can see here that obviously we're in an opening in the forest. So here's another little bit of a level of complexity, which is that, of course, even closed canopy forest, which will be fairly shaded in the understory are going to have openings because of weather events or because trees fall down. And so these are fine scale variations that we try to take into account when thinking about paleoecological reconstruction. Now, the biggest challenge really is to understand the physical structure, the physiognomy of the vegetation surrounding your site. And that is because there's a continuum, of course. Ecologists and botanists have to make these classification systems, but in reality, you can go in a continuum from wettest, most seasonally wet and very small dry season, so very continuously wet, warm environments where you have dense, diverse, rich tropical forest, all the way down to the driest of areas where, as you go, the vegetation structure gets shorter and shorter and shorter and more and more and more open. So this is something also that is our challenge, and we have to keep it in mind. Okay, now going back to the slideshow. Yes. Okay, so I wanted to let you know about that resource, and we're going to keep these things in mind as we go forward. All right. So now a little bit about taphonomy. Here's a nice illustration of lacustrine or lake environment pollen taphonomy. And so with pollen, you have differences among species in productivity. So here in this example, we see larch. That larch tree is producing very small amounts of pollen represented by these squares. So there's very little larch pollen in the air, whereas pine produces copious amounts of pollen represented by these ovoid shapes, and there's lots of pine pollen in the air. And yet there's not a huge difference between the number of larch and the number of pine trees. And moreover, larch is standing right here next to the water and pine is not. And yet it's producing a lot of pollen going into this lake. So there's differential productivity, there's differential transport of pollen. So it can come from very far away via the wind. And of course, it could also be very local. And something not shown in the illustration is the fact that the larger the basin, the larger the depositional basin, the greater the source area is for pollen, the more opportunity there is for long distance transport into the basin. So size of the ancient lake basin is important too. Then your pollen settles down to the bottom in your sediments, and then you are the researcher and you're going to count your pollen slides, all of the pollen and all of the spores, and you may just by happenstance not find, for example, in this example here, you may not find any larch pollen because your sample is swamped by other things, including pine. So pine is very abundant. Grass produces lots of pollen too. So in East Africa, palynologists working in the Pliocene and the Pleistocene, where grass is very common, have to count probably thousands of grains of grass before they get a real feel for the other plants in the community because it's swamped out. So the last thing I'm going to say is that there is low taxonomic resolution. So that is, this varies from plant family to plant family. So in grasses, a grass pollen grain is the same. It looks the same. The morphology is the same, no matter what species you're looking at across many thousands of grass species. You can't even tell subfamily. So you're not going to be able to say anything about C3 or C4 grasses or anything like that from pollen by itself. In other cases, yes, you can tell a pollen grain of oak, which is now to genus. So there's a variety of issues with taxonomic resolution when you're talking about pollen. For plant macrofossils into lakes, the key to take home when thinking about macrofossils is that they're local. kind of like all politics is local, but all plant macrofossils are local. They're not going to be derived from way far away, many, many kilometers away, like pollen. So you get a different source area when you're using pollen as to when you're using, compared with macrofossils. So the macrofossils will find their way into a depositional basin. And then after that, the key for the researcher is to hope that your fossil sediments have not been oxidized, because oxidation is the enemy of organic matter. So if the lake basin has oxygen there and you have, you have all kinds of decomposers down there, then both chemically and then the biological interactions are going to be such that your organic matter is not going to be preserved. This can also have an effect on pollen, but plant macrofossils are very vulnerable to this. These are just 3 different kinds of depositional basins, of course, from the North Temperate to Arctic areas, because they're usually more commonly represented in books and papers. But the same goes for the tropics. And in the tropics, there's the additional challenge of organic matter in the wet, warm places decomposing quite quickly. So macrofossils are local. All right, so now to talk about our project. Our Ethiopia project has been going on now for quite a long time. Originally, I started working there because I was invited by John Kappelman, who's shown here. He's a paleoanthropologist who many of you may know at the University of Texas in Austin. And he started working in the late Oligocene in a place called Chilga, which is where I started. And over the years, we've just built this wonderful team of people. This is Ellen Carano, who is another paleoecologist, paleobotanist. She also does work on plant-insect interactions by looking at insect damage on leaves. Mulugeta Faseha, who is a geologist who's now the head of the ARCCH in Ethiopia. That's the agency that oversees all foreign researchers in the country. And Mulugeta brought us to the Mush locality, the early Miocene Mush locality, and asked if we'd be interested in collaborating. And it was a beautiful find. Aaron Pan is a former student who still works with us on plant taxonomy and biogeography. Friedrich Grimson is at the University of Vienna, does pollen taxonomy and systematics. Here's Matt Clemens, who is a student of Louis Jacobs' invertebrate paleontology. He worked on the frogs. And then we have Rosemary Bush and Francesca McInerney, who has done the geochemistry of the plant-specific compounds, the carbon isotopic geochemistry. And then Teki Tesfamichael, who was a former student and worked on ancient atmospheric CO2 concentration based upon these plant fossils. And he is now a professor at Addis Ababa University. Michael Engel at the University of Kansas is a paleoentomologist. And Neil Tabor is a isotope geochemist and a stratigrapher. He did a lot of detailed stratigraphy at the Miocene site, Mush, and is at SMU, and our former student, Dan Danahy, who did palynology. And of course, there are others. So here we are looking at a digital elevation model for the Horn of Africa. And we see the thing that's prominent here, of course, are the Ethiopian highlands. And that consists of the plateau, the Ethiopian plateau flood basalts, which emerged in at about 30 million years ago, which is the mid-Oligocene. And that was before the Arabian Peninsula split from Africa. So it was Afro-Arabia in those days. And it was also before the actual development of this main Ethiopian rift, which didn't happen until about 14 million years ago. So we have the plateau basalts, imagine in your mind's eye. Now Arabia is connected. It's all one big thing. We have this plateau there of some unknown elevation. The basalts are about 2,000 meters thick. And now superimposed on top of the plateau are these volcanoes, which you can actually see in this illustration. These bumps all over the surface are Miocene age volcanoes. So they are early Miocene, and again, before the actual development of the main rift basin. So there is Chilga, the Oligocene site, late Oligocene, and there is Mush, and we have radiometric dates because they were both associated with volcanic sediments. And so we have good dates on those, both argon-argon from Chilga and also uranium-lead. Okay, so a lot happened between those two times, Chilga being the late Oligocene, Mush being the early Miocene. Between those two times, you had the connection of Africa as an island continent to Eurasia. And that created a land bridge, which then resulted in a big faunal turnover. So the fauna and to some degree the flora were evolving in isolation on this landmass that had been separated from South America and the rest of Gondwana since at least 100 million years ago. And then this connection happened around, let's say, 24 million years ago. So a big faunal turnover happened. We had warming between the two times. So we want to know, is that related in some way, or is it correlated with any change in atmospheric CO2 concentration? There's this interesting story about palms, and I'll talk about that in a moment. And we wanted to know what we could say about pre-rift biota overall. Now, to visualize this geographic change that took place across the boundary, we have the late Oligocene here. These maps are from Chris Scotise's paleo maps. And so, yeah, Afro-Arabia is still an island, although it's pretty close to these, all these islands that will eventually become Europe. And then when you cross over into the Neogene and the early Miocene, you can see that there's this land connection here, and that's the route for animals crossing in. And there's this incipient rifting developing. So of course there's tectonism going on and places where sediments can accumulate in basins. All the dark symbols are other paleobotanical sites of various kinds. Sometimes just isolated pieces of wood or an isolated seed or fruit. Yeah. So how does this all fit into the bigger context of importance? In addition to the climate change issue, there are some really interesting questions about biogeography. This is the modern distribution of the African tropical forest blocks. So you see that they're Central Africa, West Africa, Eastern Africa, and that they're separated. With this, of course, being the corridor again, that's much drier because of the Rift Valley. So early on, it was thought that, well, there's the rift. If the rift came along 14 million years ago, then the separation of these, and we are imagining one continuous forest must have happened 14 million years ago. And since that time, you've had evolution happening. And so therefore, sister taxa here and here have been evolving separately for 14 million years. But the story actually, of course, as always, once you start getting into the details, is more complicated. And it turns out that probably different plant families, different groups have different evolutionary histories depending upon circumstances. And so, for example, this plant family, the Ananaceae, study of its phylogenetic analyses found originations happening at these various times in the Cenozoic. So the authors, Couvreur et al., proposed several times a forest breakup and reunification related to climate change. So there's really interesting things to try and sort out here. Then species richness is another really interesting story about Africa on the whole. Tropical Africa is relatively species poor compared to tropical South America, tropical Southeast Asia. And I think perhaps we're just beginning to try and get a handle on why that is the case, because when Africa was united with South America, of course, they started from the same place. Their plant communities were one. So why is South America so much more diverse than Africa since they separated? Well, there were some different things that happened in each of those places that caused differences in climate evolution. But just to give you an idea of how different they are, we see here something called Fisher's alpha. It's a measure of richness. And so just as a comparison, just to give you relative numbers, the highest Fisher's alpha that you can find among modern sites in West Africa is between 41 and 66, whereas in South America, the richest sites have values between 151 to 300. And that's because the very richest sites in South America, or at least I think this is why, the very richest sites in South America are in places where you have high aseasonal rainfall and topographic heterogeneity. So there are a lot of niches into which species can evolve. And I think that Africa doesn't have that combination of characters to get its maximum species richness to the same levels as you find elsewhere. It's very interesting. In South America, the palms are like the poster child for this idea of differences in richness. So Africa has a total of 65 species of palm, but South America has 451. And then Indo-Malaysia and Australasia also have hundreds of species. So palms are really, again, interesting and kind of special. And I'll just mention right here that Chilga, the late Oligocene site, is full of palms. Everywhere you go to sample, you have palms. So palms were, several subfamilies of palms are represented among those. And the ones we've identified so far still occur in Africa, but to a very limited extent. And the palms were obviously really important in an ecological way in the late Oligocene. In the Miocene, we have one palm that shows up in the pollen, but none in the macrofossils. So, and then paleoclimate, this is what happens across the boundary. We have Chilga shown here. This is the Zakos curve, and which is based upon oxygen isotopes from benthic foraminifera. And so you have Chilga here at about 27, and you have Mush here at almost 22, and you see that there's a warming between one and the other. So it's not a huge warming, it's not a kind of catastrophic warming, but it's a warming perhaps on the same order of magnitude as the warming that we are seeing in the 21st century. So what does that mean, where we have these two sites from the same general region, and what is that going to show us with regard to atmospheric CO2 concentration? So the research objectives to document the pre-rift vegetation, to demonstrate the potential for a multi-proxy analysis for getting really high resolution understanding of the ancient environment. And then we wanted to examine the sensitivity of tropical forest ecosystems to environmental perturbations at temporal resolution comparable to Quaternary pollen records. And where that comes from is the lake sediments that I'm going to talk about from the early Miocene probably represent a time interval of about 50 to 60,000 years. And that's based upon comparison with known long cores from Quaternary sediments and their age estimates. So cores that have good time control, how much time is represented on average from place to place in different places around the world. And so what we came up with is this rough estimate of about 50 to 60,000 years represented. And in Quaternary studies, when you look at that kind of a time sequence, you will go perhaps from the Pleistocene into the Holocene, where everywhere around the world you see a major change in vegetation. What are we going to see here in this environment? So this is the Mush locality looking down on it from the top of the plateau, and the Mush stream comes down in here and then goes all the way eventually out into other tributaries of the Blue Nile. It's always running water. It's lovely. It's fed by a spring and looks like this close up. All of these sediments are carbon rich. So these are carbonaceous shales that you can then, crack apart. But you can see that in the summer rainy season, the water must be just roaring down here. We can't get up here in the rainy season. These boulders are huge for a reason. And we would have to sort of reorient ourselves every year when we came in the dry season, because the beds would be eroded and look slightly different each year. The first time we went out there with Mulugeta, we were amazed by the kinds of plant fossils and the abundance of plant fossils we found. So these are all leaves, whole leaves or parts of leaves in this organic rich sediment. And here is Mulugeta enjoying himself with finding some more fossils with us. So below him, you can see this beautiful big ash bed. And so we were able to get good dates on the ashes below, just underlying the beginning of lacustrine sedimentation. This is the stratigraphic section in detail put together by Neil Tabor, and this shows the location of that big ash bed and the date shown here. We have 7 meters of section that we sampled vertically, and we just refer to them as levels A through F. So A, B, C, D, E, F. I don't know if you can see my pointer. If not, look for A, B, C, D, E, F. And then the 7 meters, as I said, represents about 50,000 years. We have plant and insect herbivore damage censuses that we did. And we also sampled compound-specific organic geochemical signatures. So for example, these chains of carbon, these molecules are called N-alkanes, and different chain lengths represent different parts of the biota, and can tell you can actually get delta C13 values from the different alkanes, the different chain lengths. So that was interesting. And then each of our censuses comprises 1 to 3 centuries of sedimentation. And we also have 6 productive phytolith samples, so from 6 different layers. And here they are shown here. These pie charts are the phytolith samples. We also have some pollen from level B. There is an ash bed at level C. So I'll refer to that again, because we don't have it's not very rich in diversity, because we don't have a very big sample, because it's actually an ash. Okay, so our sampling scheme is like this. We have alpha diversity is represented by each of the quarries that we collected at one stratigraphic level. And there they are from levels A through F. And you can see that level C, the N, which is the number of leaves, is low. All the other quarries have well over 100 leaves. So C has to be ignored in some cases for our analyses because of the small sample size. Okay, so alpha is each quarry, beta within diversity. So that means within each stratigraphic level time unit. And then we did beta diversity between levels, and then the total diversity of the whole site. So these diversity measures are to compare and understand species richness at different levels, and how heterogeneous our floras are. So do you leap from, say, a low level of diversity at the quarry scale and at the level scale, and then suddenly you jump up to huge diversity when you sample another stratigraphic horizon? That's what this is to assess. This is how we did our quarrying. So this is a bench quarry, and here we are getting out these blocks. Ellen, sitting there on the left, is holding a block. We tried to get them out as big as possible. Each unit, each collection, each block was only 5 centimeters thick. So we followed the same horizon from quarry to quarry till we had 3. In one case, we had 2. Just to give you an idea of what we found, a little bit of what we found, there are a total of about 50 taxa from here. And we have, over 2,000 leaf specimens. We also have some fruits, seeds, and insects. Okay, so we have just examples. This is one of our unknowns. This one here is called Taka, which is an understory forest plant. We have aquatic fern in one horizon, which is called Salvinia. Here it is, here just to the left of that picture of Salvinia. The leaves are beautifully preserved. They're compressions, so they were just pressed. They landed down at the bottom, which seems to have been anoxic, and so they did not decompose. And every layer of leaves is they're just layers of compressions. We have the microscopic leaf epidermal characters, so the cell outlines of both the stomata, and on the upper surface, usually there's no stomata, but we have the cell outlines, and including little things like leaf hairs and glands and things like that. Those are valuable for telling us when we have the same taxon. If we're looking at 2 leaves, we're not sure if they're the same or different. We can look at their cuticle and say yes or no. And also because cuticle characteristics are pretty conserved, and so it can help you relate to a living relative. On the left is the palm genus Sclerosperma, which is what was found in the pollen samples. And that is the only place we found Sclerosperma. And today is restricted to the low-lying forest, wet forest communities. And oftentimes it's in swamps. It's actually called a swamp palm. Here's another thing that we found a lot of, which is the seeds of this plant called Newtonia. And to the right, you'll see the distribution of Newtonia today, with mush shown in Ethiopia there. And this pattern, this modern distribution pattern, is seen again and again and again and again among our identified fossils from both the Miocene site and also at the Oligocene site. So this is the distribution that mirrors the forest communities today. Newtonia is a legume. So these are the seeds that come out of the pods. They're really cool. Here's our most common taxon, which is called, we now know as Anglerodendron. And it's also a legume, beautifully preserved. You can see the stomata and the cell outlines. Here's a comparison in a recent paper on the paleoecology at MUSH, showing first to the left there is legume 1 that I just showed you a color picture of. The second one is another legume, which is very closely related, actually, called Anthanotha. And then the third one on the right, the pear, that one is still referred to as interrupt. That's our pet name for it, because we don't have an ID on it yet. But these are all legumes. The whole flora is really dominated by them, especially what we used to call legume 1. It's the most common. And this is not unusual for tropical wet forests. So this is what I'm leading towards. Okay, so here is Anglerodendron shown on a herbarium sheet. And here's the Anthanotha, which I just showed you, and you can see its pods hanging down there. Another pollen genus is Hygenia. Hygenia is a plant that many of you may have heard of if you've worked in Ethiopia. It's associated today only in the highlands, in the high forests throughout the shoulders of the rift and the mountains, the highlands of Eastern Africa. And we have it at MUSH. So does this mean that MUSH is a high elevation forest? Well, we don't quite know, because they have other things like Newtonia, for example. And because also with climate change, with a different climate, what's going on here? And what's probably going on, and I'll get to this again, is it's not an analog. There is no analog to this today. But going on, this is the Sclerosperma, the swamp palm pollen, beautiful, gorgeous imagery from Friddy. So these are done with a field emission electron microscope. So now what does the whole flora look like from level A to level F? These are relative abundances of our most abundant taxa. And you can see that legume 1, which we now call Anglerodendron, is the most abundant throughout the section, except for level A, where it occurs in lower numbers. And since these are relative percentages, as this is a lower relative percentage, it allows for an additional presence, basically, or greater presence in our counts of other taxa in level A. How do they sort out relative to each other? Are they very similar? Are they very different? When we do non-metric multidimensional scaling, which is just, it places your samples, in this case, it's placing our quarry collections onto a plot that visualizes how close how similar they are to one another in terms of the species occurrence and species abundances. Yeah. So you see here that on the left side, the quarries from level A, well, first of all, the quarries all plot nearest to each other because there's not that much difference between one quarry and the next one. They're only separated by about 10 or 20 meters. The levels, quarry from the levels, all seem to overlap except for level A and level C. C, we can set aside because it's a very small sample size. A, though, is different because it has a low count of that legume 1, Anglerodendron. So it then has different relative percentages of all the other taxa, and it makes it a little bit different on this plot. Otherwise, they're all very similar. These are a lot of numbers and writing, and I don't want to have you read all this, but I just want to point out a few things. So here we have all our levels, and these are attributes of the different levels in terms of floral diversity and evenness, which you can measure. And in modern ecology, you would be doing that for your species counts and your species lists. And here we have to remember and not forget that we're doing counts of leaves and for each of the species, not individual plants. But one thing to point out again is that there is a big difference in the percent of leaves that are legume 1 or that Anglerodendron, and I've circled that in the upper table to the left, so 30.9 at level A and well over 50% and as high as 65.8% of all leaves from a particular level. And the richness values, the other values are all fairly similar as well, perhaps with the biggest difference being either level C, which we, again, have to take with a grain of salt, or level A. Okay. Similarities, again, we know this from the multidimensional scaling, but we can see it here using Jacquard values. And so that means that more than 50%, we can think of it that way, of the taxa from level to level are shared, except level A. Okay, that's 39%. All right, we also have phytoliths, which is wonderful. And I should have put a picture there of Alice Novello, who was a student of Doris Barboni, who they both worked in Africa quite a bit. And she came and worked with me, and she also did a postdoc with Caroline Stromberg, who's done phytoliths all around the world. And so the phytolith information is really interesting. Here it is compared with modern to understand what the phytoliths are telling us. And the basic conclusion is that it's telling us there is forest, lots of forest present there. There's almost no grass at all in the mush assemblages that produced phytoliths. One really cool and interesting thing is that among these phytoliths is one in particular that is only found among a small subfamily of grasses that only occur in the understory of wet forests. So for example, this taxon occurs in the lowland rainforests of, I think the farthest east it goes is Uganda. So that's kind of cool, because also I found that one as a leaf in the middle Miocene in Kenya in a forest assemblage. All right, mean annual precipitation, we get that from leaf area calculations. So the bigger the leaves, the greater the rainfall. And I did a study where we related the leaf areas of different plant communities to rainfall in those different plant communities. So the reconstructions from A to F are here. We can actually look at, whoops, I put that around the wrong column, darn it. One column to the left is the mean annual precipitation. So you can see it goes from about 1647 or 1597 to 15 something. So these rainfall values are all very similar. Their margins of error overlap and they don't show very much difference. For temperature, we have to use the overlapping distributions of the modern relatives of our fossils. And we're just getting to that for mush. Aaron Pan is working on that. So far he's plotted out these two taxa. And again, you can see that mush is up in Ethiopia. You see where the star is. The circles and the dots and the triangles represent these two taxa that occur at mush. And we can see again that same pattern. The modern equivalents, the modern relatives of our fossils occur in the rainforest biome, the lowland, moist, broadleafed tropical forest biome. Yeah. And those, by the way, those ones that occur in Eastern Africa in that corridor are up in highlands, so they get more moisture that way. What about PCO2? So this is Teki Tesfamichael's study. He looked at this one taxon from the mush locality. The cuticles and the detail on the cuticles is amazing. You can just, it's beautiful to see the images of these plants. And so to get the partial pressure of atmospheric CO2, he used a gas exchange model that was published by Franks et al. In 2014. Not enough time to go into details about the model, but it involves measuring the stomatal openings so that that will help to determine how faithful the CO2 coming in is going to be to the general atmospheric CO2 concentration. And then you measure the delta C13 values of the plant and a few other things that are given in the model. And you can then produce estimates of ancient PCO2. So this is how Teki's samples plot out in a paper that was published in 2017. So for Chilga, we have values that are almost like today. And then in the early Miocene, we have values that are higher than at Chilga. And if you remember, it was the temperature got higher at Chilga. So there does seem to be a positive correlation between atmospheric CO2 concentration and temperature, mean annual temperature. And these are some other publications that have greater ranges of values. Their dates also are less precise than the dates that we have. So there is that benefit in this case. And then I think this is finally here, the results of the geochemical analyses that come from the organic matter itself. And so here it's a little bit confusing, but I'll explain it that these different C numbers, C23, C25, C27, refer to different chain lengths of these carbon chains that are called alkanes. So they're N-alkanes because the N value, the number of carbons in the chain varies. So the long chain lengths like C27, C29, C31, these are usually C29, C31 especially, are associated usually with higher plants, flowering plants. So those would be from the leaves contributing to the organic matter in the lake that come from the forest next to the basin. And their values are, all the values are fairly negative values, and they indicate that there's basically, we don't find any evidence of any warm climate carbon isotopic values. So in other words, no C4 vegetation. Whoops. One interesting thing that came out of these studies is that some of our N-alkane values are extremely negative, and they come from a compound called hopanes, and hopanes with extremely negative values are associated with methanogens or anaerobic bacteria, and that's what you would find, of course, in a substrate that has no oxygen, and so that kind of fits with our idea as to why we have this incredibly beautiful preservation. So going back to our research objectives to round it out now, we know we have forest vegetation at both Chilga and Mush. The Mush forest may be actually the first example in the fossil record of what is called a monodominant tropical moist forest. So most tropical rainforests around the world are a mixture of species, and one may be more abundant than the rest, but it's not a monodominant. In other words, it's not one species that dominates the whole thing. In Africa, there are more monodominant forests in the tropical forest, the wet forests, than elsewhere in the world. Today, those are dominated by close relatives of Anglerodendron, which is what we have being tremendously abundant. And in comparison with leaf collections from these monodominant forests, it seems that levels above level A, because level A doesn't have as much of the legume one or Anglerodendron, we have this probably a monodominant forest, and so that's pretty fun. Have we demonstrated the potential for multi-proxy analyses to provide some kind of robust idea of what was there? I think we can say yes. And what about the sensitivity to change? So within the 50,000 years represented, we hardly have any change at all. And so this seems to represent a very stable kind of plant community, perhaps with the transition from level A to the upper levels, from a mixed forest to a monodominant tropical wet forest. But it basically represents ecological dynamic equilibrium, which is very interesting. We need paleo temperature estimates from paleosol oxygen isotopes, and we are getting those. We're waiting for those. I'm going to fix my plug in my computer, I'm sorry. And we need a means to estimate paleo altitude, and we can talk about this more if you want to. I think I'm probably pressed for time. And so that is all I have, and I'd love to take questions if you have any.
Wonderful, Bonnie. Thank you so much. I know
Yes.
you may have an event coming up at the hour.
No, I changed it, so I'm okay. I'm good to go.
Okay, good. So we have some questions for you.
Good.
Thank you. That was wonderful.
You're welcome.
Very, very, very interesting presentation.
Thanks.
And,
Thank you.
Yeah, that looks like an amazing site, and the comparison to Chilga, I think, is very, very enlightening. So, first question we have here. So Chilga now, Mush in the Ethiopian highlands, of course, is in a very different setting today from what it would have been in the early Miocene. We don't know about the elevation, but it looks like it would have been a relatively high area. But what about latitude? As you've shown, Africa has been moving sort of north, and way back in the early Miocene, the site would have been farther south latitudinally.
Not very much, though.
Very much.
Yeah,
Okay.
I think it's pretty much. Yeah, I think it's pretty much where it was. So I would say it would have little to no impact because of the tiny amount that it would have changed. But what's probably more impactful than latitude would be, of course, precipitation patterns and topographic features that maybe we, need to take into account with modeling, perhaps. Yeah.
And
Oops. I think you're at.
yep, it looks like from your data that except for these grasses that would have been present in probably tropical rainforest conditions, you don't have much in the way of grasses. Is that correct? Um, but you mentioned also that of course we need to distinguish between a C4 grass that one can detect with carbonates. Um, what is there evidence in Mush or elsewhere from this time period of C3 grasses in some abundance? Just thinking about what these landscapes would have looked like overall.
Oh, that's such a great question. Yeah, so interestingly, at Chilga, so the late Oligocene, so Dan Danahy, who did his master's on the pollen, found, I don't know, maybe up to 10%. And also, actually, Kedi Yamani in the past Yamani and Bonfi published some pollen work from Chilga. So there is evidence of enough grass that you get the grass pollen in there in noticeable percentages, nothing huge like you would get in Eastern Africa now. So, and there's nothing to tell us from the isotopes that you have C4, so then those obviously have to be C3. And the question I always wonder is, are these grasses associated with wet areas? So are they just aquatic, or grasses that are water loving, or are they grasses from, out on the landscape? And I would say, I think we should at least consider, or perhaps even assume, that some of them are associated with the bodies of water, because you have to have some kind of depositional setting to have the fossils. And so that's why we usually find things also mixed in with our terrestrial things. We find things that are associated, with water. Now, at other sites, this is really interesting. And I know that I'm not giving anything away because talks have been presented at GSA and other places. And I know that there's a paper one or two papers in the works, but from Uganda, from Moroto and from Rusinga, there seem to be some open areas. And not only would we find C3 grasses, but there's evidence of C4, even as early as the early Miocene, which is amazing. I mean, that's big news. And the reason that it's so, the reason that they've concluded that is not just because of the carbon isotopes, but also because there are phytoliths now. So they can link the phytoliths to the carbon isotopes. And when you can do that together, then you have a more solid footing when you're going to say something big like there were C4 grasses in the early Miocene. So it's a little bit younger than Mush, but I think my personal view is that it's because of the difference in altitude. I just think that, this place, Mush, is capturing rainfall because it's higher enough. We don't know how high, but maybe higher enough than the Western Kenyan and Eastern Ugandan sites. Yeah. So that shows heterogeneity, on a really big scale, like big time scale and big spatial scale.
Yes, I think in Kenya and Uganda, so these sites would be a couple of a million years younger overall.
Mm-hmm. Yeah. Very frustrating not to have the same age at both places.
But you made a comparison there also of Chilga with Mush, and you have faunal turnover I think you showed a picture of maybe Arsinoitherium or something like this.
Mhm.
Um, Now, at Mush, you don't have mammalian fauna, right?
Yeah, and I didn't take the time to show the fun frogs, but well, we do have mammals. We do. I shouldn't say we don't have any. And I'm glad you brought it up because you and I talked about it yesterday and I forgot to stick it in. But we do have a few specimens. Lewis found Diamantomes, which I'm proud to remember the name of, which is a rodent that is also found at places like Rusinga. So it's a typical early Miocene kind of a small mammal. We have crocodiles, which don't tell us much. We have a anthracothere tusk. It's very small. It's about this big. And again, so I think we can't, say something more specific about it. And then one or two longer bones that you can't say what it's from. So the bones and even these are in pretty poor shape because I think the reason that they were there is that they were probably associated with concretions and were protected in that way by the concretions. So I suppose if someone had the energy, they could maybe go back and just sit there wailing away on concretions for many hours and see if there's anything more. We did open several, but we didn't find it. We weren't lucky in finding bones. Um, yeah, so that's it, except for the frogs, which are compressions, just like the plants in the lake beds. They're just squash flat. You see the tadpoles, you see when they have bones, the bones are there. You see everything, all the soft tissue. Yeah.
You mentioned the Kenya and Uganda sites, uh, but if one were to think about the whole continent at the time period of Mush, give or take a million years or even a couple of million years, can we get a picture of uh, continental scale vegetation, do you think, or is that just too difficult at the moment given the samples that exist?
Well, we we can yeah, we can probably speculate in a somewhat educated way. And so the big difference when thinking, of course, about Eastern and Western Africa is that in Eastern Africa, you have so much more topography that came about. Of course, it started clearly in the early Miocene, at least, if not the late Oligocene, because you had to have the tectonism forming these little basins to have the fossils. But then you got this big rift. So we just confine ourselves to the early Miocene. I think if I were to make a video in my head of what's going on as you transition from the Oligocene to the Miocene, I would visualize for a lot of forest. I mean, a lot of forest, even across the board in the Oligocene. Well, across the board is a little bit much, but let's just say lots of forest at the lower latitudes. Variations on the scale of the variations we see now, given, just local differences in edaphic settings, maybe wetter places, drier places in the soil there. And many precursors to the genera that we find today, because we're finding a lot of genera that exist today in Africa in forest communities. And then as you get to East Africa, as you get into some topography, you start to differentiate. And as the movie goes from the latest Oligocene to the early Miocene, it would be wonderful to actually make a movie showing the faunal change, with you have the classic, bovids eventually come in, and well, you know this very well. Bovids eventually come in, you get giraffes, you get true carnivores, and the arsinotheres, the poor things, are lacking in whatever it is they need. And so other things go by the wayside, and and elephants, which are there from before, may decline in diversity, but you still have dinotheres, you still have gomphotheres, right? And you have and hyraxes. So you have that going on in a heterogeneous big landscape. And as the rift develops, you have to have vegetation that is adapted to drier climates coming in, and it's gradual and it's complex. So what used to be thought was that at 14 million, you had the rift develop, and boom, you went from all forest to modern. And of course, it didn't go like that, because there's a site in the Tugan Hills that's 12.5 million years old, so post-rift, and it's a forest, and it's a forest with affinities to Ugandan forest. So yeah, lots of variation in Eastern Africa through time. Yeah.
I mean, with the rifting, I suppose you get more heterogeneity.
Yeah.
And that, some of that is probably the breaking up of extensive rainforest. But as you mentioned, that's going to depend a lot on topography, and you may get some areas with higher precipitation because of rifting, and
Exactly.
even more woody vegetation.
Yeah.
But on that line, I wanted to ask you a question. You mentioned the species richness in South America compared to Africa. And in the diagrams that you showed, I think one can see that the very high richness, and I think you mentioned this, is associated with what's the eastern part of the Andes.
Yeah.
So you get both rainfall because of the Andes, but also topographic gradients. So the Andes, in a way, would play a key role in that very high diversity in South America. Would you say that that's correct? And if so, why didn't the Rift Valley and its elevation, its topography, elevation gradients, play a similar role in Africa in terms of enhancing the richness of plants and animals?
Right. So, right, we did. Okay, so this question has a couple of answers. So in South America, it does seem that today that is where the highest diversity is on the eastern side of the Andes. You have the combination of the rainfall, the aseasonality. That's really key. So it's high rainfall combined with low seasonality and topography. And you don't get that combination in Eastern Africa because the seasonality and the rainfall just don't get there. So at Chilga today, for example, Gondar has a dry season of about 7 months. And so in the Andes, you have a dry season of 0 to 1 or 2 months, maybe. And in places in Africa where you have a dry season of only 1 or 2 months, you're down mainly in like the Congo, Eastern Congo, all the way to the west. So, but then, yeah, so that's what the hypothesis is right now. I mean, I can't say this with 100% certainty. That's the hypothesis. But we looked at whether the decline in richness, and I mean, all we have to look at in great detail is comparing Chilga to Mush right now. So we did do that. And it seems that Chilga there are sites there that are really, really diverse, really high diversity. And there are indications that there was less of a dry season. So, right. So Aaron, my former student, did a map of the distributions of modern genera, 10 modern genera that he identified at Chilga. And they all overlap, 8 to 10 of those overlap in their distributions today in places that have relatively short dry seasons. And those are things that don't occur in Ethiopia now. So it seems that it's the impact of the dry season that's limiting their distribution. But it's the collection of all of them together where we're trying to estimate total diversity compared to other things. And it seems to us that one of our main sites in Chilga is really highly diverse, much more diverse than we find at the Mush locality. So it's possible that, that sort of represents some ancient level of diversity that we don't sample elsewhere. I know that the pollen records from West Africa that were done ages ago by Szilárd Sebeldaev is the name of the first author. She did long cores, very long cores from West Africa, and also studies by Morley, who reviewed the vegetation evolution in Africa. They also show very high diversity early on and then declines at, say, the Eocene-Oligocene boundary and again at the Oligocene-Miocene boundary. So perhaps that's related to this latitudinal shift that takes place at that time scale, going from Eocene all the way north. But the Eocene is tricky too because it's fairly diverse. It's diversifying in West Africa. In Tanzania, where I did a study of Middle Eocene macrofossils, that paleoflora is crazy. It's very dry. It's dominated by legumes. It probably looks something like a Miambo woodland, but the taxa are all different. So maybe because of intense heat. I don't know.
Could that have to do with just the fact that it's more to the interior of the continent?
Possibly, yeah, because it's true that those pollen records on the West Coast come from coastal areas, but also by the Middle Eocene. I have to say that there are hints of grassy areas, but again, we don't know if they're really just aquatic grasses or not. So I'm really out on a limb here. I'm about to cut myself off the limb because we don't have enough sites, but these are kind of things that I'm thinking about. These are speculative things yeah.
So you gave us a very nice image in our minds of moving from Oligocene forest through the early Miocene, and then you mentioned a more complex breaking
Yeah.
up of forest through the Miocene. And the question is, as you know, there's so much about early human evolution now, and earliest hominins, and even just before the earliest hominins, possible relationship with wooded environments, forest or woodlands. Um, what's your view in terms of is there a persistent of proper rainforest elements in Eastern Africa way into the late Miocene, or are these not rainforest but woodlands of some kind of another, and probably grasslands as well? But are there elements at least, or at least some regions that may retain tropical rainforests as we move into the late Miocene in Eastern Africa and some of these places where we lose our I'm sure that that of course in Central Africa and West Africa we would have had persistent rainforests, but
Right.
would that be the case at all towards the east?
Well, interestingly, so when you get to about 10 million, there is Nakali. There's a small collection of plants from Nakali, which is difficult to deal with because they're just leaf impressions and they're not great, in terms of preservation. So we have no organic matter. But they do look like woodland to me, woodland to forest. And there's a site in the Tugan Hills that could be somewhat similar in age. I don't know, the Nakali site is dated by just stratigraphic correlation to other places where they understand the chronology. And it reminds me of some of the plants from the 12.5 million year old site. And that site is definitely a forest. I mean, I would say it's a forest, and of course, as I said earlier on, we have to be careful because, forest means taller trees and overlapping crowns, and it's difficult to pinpoint that. But from the taxonomic list, I think it sounds like forest, very similar to Ugandan forest. So, so maybe as late as 10 million, but everything I can think of that comes from the late Miocene, you really start to get grassland expansion in a big way. And I don't know, we don't have examples of forests, like real rainforest hanging on to the latest Miocene, but that could be we don't have the sites, I do know that there is, this goes into the Pliocene, where Raymond de Bonfils reported Antrocaryon, which is a typical forest. I think it was a seed or a fruit from Hadar at about 3 point something. Do you remember that? And I think later she told me it was a misidentification. Am I right?
I'm not sure about the misidentification. I think there is a report of Antrocaryon from Omo, from the Omo.
Okay, Omo. Okay.
And that, along with some of the invertebrates, have led people
Right.
to think of the Omo, at least at times in the Pliocene, not into the Pleistocene, but at least in the Pliocene, um, being quite forested. I mean, not just a woodland, but closer to a forest. But I don't recall seeing the correction about a misidentification of the.
I probably, maybe I shouldn't say that. I should ask her. I should ask her again to clarify. We were just chatting one time, so um, but this brings up another point, which is interesting, and that is that we're thinking of it as a progressive loss of forest, but in the Pliocene, the mid-Pliocene, which is approximately that period of time, there was global warming, and so it could have been wetter, and that could have supported the reemergence of some forests in some communities. So that's possible, yeah.
Yeah, we've been working on the Morsi formation of the Omo with Michel Drapeau and John Wynne and others in the last
Good.
few years. And there, I'm not sure I would describe it as a forest, but by comparison to other sites in Eastern Africa of similar age, it looks like a much more closed, wooded environment, much more than anything else that we've seen based on different lines of evidence. But again, I'm not sure if we can call it a proper forest or just a heavily wooded area with grasses in the distance far away.
Yeah, and this is, sometimes even the best description you can give, even for a modern place you're standing right in. You know what I mean? Because it's hard to sometimes really characterize these places. Is there any organic matter preserved? I'm curious.
Well, John Wynne he has a paper now looking at the carbonates. So we do have carbonates
Right.
and stable isotopes, and they look very interesting. I think there's a paper in review now, so
Good.
we look forward to seeing the results of that. But it is I think a really interesting site just because of the contrast with places like Canopoy farther to the south,
Mhm.
or even the sites in the Afar that are of that time period, Asaisi and some of those.
Yeah. And again, that's a perfect example of where it's possible. Maybe we don't know or maybe you do know better what the paleotopography was in the wetter place versus the Afar, which by then must have been, pretty darn dry. Yeah.
But yeah, so in the Mursi Formation, for example, even today, when you're there, at times it's a very wet place, or can be a very wet place, very heavily vegetated, not a forest, but very thick vegetation. And then you go a little bit, 100, kilometers or so to the south, and you're in the Turkana Basin per se, and it's completely dry, no vegetation except in the lagas and so on. But and some of that has to do with precipitation in the Ethiopian highlands. That some of that certainly gets to the lower Omo, to the Mursi area, but not so much to the as you go south to the Turkana Basin.
Yeah. Yeah, it's really complicated on a, you know, that's even 100 kilometers, that is a small scale, you know, in terms of what we're thinking, we're trying to piece together. Yeah.
We have another question for you, Bonnie, about the coastal forests. And in our group, we have a special interest in the coastal forest of Eastern Africa because of the paleontological sites that we have in Gorongosa National Park in Mozambique that we think we're getting samples from Miocene times of coastal woodlands. So we have fossil wood, we have marine invertebrates and vertebrates, and also terrestrial mammals. Um, but there has been this idea, and you mentioned of this separation of the main forest blocks in Central and West Africa from the eastern coastal forests. And question is that separation too, although it's very much a fact today, very likely would have happened in very complex ways. So not establishment of some kind of arid corridor, and Jonathan Kingdon has written about that, but very likely with all kinds of complexity in between and connections coming and going. And Kingdon has talked about, as you mentioned also in your talk, taxa in the eastern forest having their closest relatives in Central Africa. Is that also the case with plants? Do we see the same thing with vegetation patterns?
Yeah, that pattern is repeated. I think there's work on the plants for sure, showing connections in terms of biogeography, sister taxa between the coastal forests and the main, you know, blocks of forest in Central and West Africa. And also, I think people have worked on butterflies, looking at connections. It's really interesting. And as we know, well, one thing I should say is that there are the coastal forests, which are perhaps a little more continuous latitudinally, and then there are the Tanzanian Eastern Arc forests. I don't know if you're aware of those. So like the Usambara Mountains in Tanzania, they have an amazing, wonderful flora that is rich, and there's a lot of endemism there. Those are old mountains, but they do still have connections to Central and West Africa, and also in some cases, if you think about it broadly enough, to Asia. So there are those, and some of our fossils sometimes look or are seem to be most similar to things that you find in the east, in Eastern Arc Mountains, and also in Central and West Africa. So yes, there is a complex history. There are botanical, certainly, connections between coastal forests in Central and West, as well as coastal forests and the Eastern Arc Mountains and Central and West. And I think sorting out the biogeography is one of the most interesting things to me, because it gives you a visual of things moving around and then evolving separately. And it's really pretty interesting. So getting a site where you have a site is going to be amazing. Well, you already know, but I'm excited to hear about it.
And we will need to go soon. Just one last question
Okay.
about comparing the fossil leaves with modern leaves that look so similar. Is there some kind of conservatism that we have with plants that we don't have with animals, where you can look at a leaf from 22 million years ago and one from today, and they look almost identical?
Yeah, absolutely. It's amazing. You know, you go from, in the Pleistocene is a perfect example, because you go across the Pleistocene Holocene boundary and you have these giant extinctions happening. And in the plants, you know, you'll rarely ever find the evidence of an extinction at the end of the Pleistocene. So yeah, there is this conservatism, and I think it's because your basic flowering plant, I guess, plan, you know, the way that flowering plants reproduce, their physiological, their physical, morphological adaptations are really small and varied. They can grow everywhere, every place. So they can also move and grow in other places. So yeah, there is this conservation, and in fact, rainforest leaves in general have a sort of classic shape and structure, the leaves themselves. So, you know, they're always like, they have these drip tips and the smooth margins. And, you know, at a glance, somebody who's not used to looking at details of the veination might just say they all look exactly alike. When you look at the veination, the examples I gave, with some of those specimens, you cannot, even if you know the details of the fifth order veins, you can't tell the difference between species A and species B without looking at the cuticle morphology. You actually have to look at how the stomata are arranged and how the cells are arranged to tell that they're different. So yes, there's definitely a conservatism in plants as compared with the animals, for sure.
Well, thank you so much, Bonnie. I think we've taken a lot of your time.
Oh, I enjoyed it.
That was a wonderful talk.
Thank you.
I think we've all learned a lot, and of course, we would love to continue the conversation. And next time you're in Oxford, we can continue in a pub. But
oh, that would be wonderful.
Thank you so much. We really appreciate you taking the time to talk with us,
Sure.
and look forward to seeing you again somewhere in Eastern Africa.
I know. I run into you around the world. I'll see you someplace in some exotic place someday.
Very well. Thank you very much, Bonnie.
Thank you, Renee. Thanks for